So thank you very much for welcoming me here. I'm very honored to be here, and so I'd like to introduce myself, spend a moment introducing myself. Brett had to do a little bit of improvisation in the biography that was included in the program because of my travel schedule. So I'm just going to spend a moment introducing myself and letting you know my background. So I'm a biochemist by training. So first of all, I'm the DEB program leader at Shire, the dystrophic EB program leader. And, but I'm also a biochemist by training. And I've worked in rare diseases my entire life, career, that is. And as soon as I was done with my doctorate, I went to work for this company called TKT. And we worked on rare diseases, and TKT eventually was acquired by Shire, and I stayed on, and been there since. So I've worked on every treatment for rare diseases that we have at Shire, and many that are still in development. So with that, I'd like to also, in various capacities. So back last year when Shire decided to acquire Lotus, I was approached to become the program leader, and I was thrilled to be part of this effort. And I'm thrilled to be here, and thank you again. I'm learning a lot from all of you, and I'm learning a lot from the experts, and it's been a great conference. Okay, so now I'd like to spend just a couple of moments introducing Shire to you, because many of you may not know what Shire is, and also because Shire is a little different from some of the other companies out there in how we approach things and what we look at. So we have a focus on developing treatments for diseases that have high unmet need. And we're also focused on rare diseases, started back when we acquired TKT. In rare diseases, almost all of them have very high unmet needs, so it's quite the synergy there. Something else that I want to point out to you is that we have a lot of talent, a lot of expertise. We've been doing this, we've been working in rare diseases for over 25 years. Most of the treatments have been protein replacement therapies. And we really do believe in our logo, and it's to be as brave as the people we help. And we see courage in everything you guys do, and we try to be a little brave ourselves, sometimes doing things faster or a little different, and when we're told that something can be done, well, we don't always believe that. So we try to take a little bit of that courage in our work. Shire around the world. So it was founded in 1986 in a tiny little town in England. And it's grown through acquisitions like TKT and like Lotus, and also some products that were homegrown, let's say. We have campus and acquisitions. So Lotus was acquired, and that's very common in an industry, and it's becoming even more common these days. Shire acquired TKT, and it acquired Lotus, and it has acquired many other companies even in the last 12 months. So we have campuses in Massachusetts, in Pennsylvania, in the UK, in Switzerland. Those are our main campuses. But we reach all these countries that are in blue on this map, and probably a few more by now. And our areas of focus are four, but I'm going to just have you look at the left side, because in research, our primary focus is in rare diseases. Some of the diseases for which we have approved treatments are listed. There are many others that are in development, that is either in clinical trials or even earlier than that. The list is very long, and these are just some of them. So we also focus, when we focus on rare diseases, we focus on diseases that have known genetics. by that I mean that the gene is known, and dystrophic EB is a perfect example. Mutations in collagen 7, very well understood, the genetics. Rare diseases overwhelmingly have high unmet need, and I'd like to point to you something in this. Let's see if I can make this thing work. That together, orphan diseases impact one. Did it work? There. 1 in 10 individuals. So the work that DebRA and the Jackson Gabriel Silver Foundation and all these other wonderful patient advocacy groups does is wonderful. And also the work that, reaching out to other patient organizations in rare diseases, and together, there's quite a bit of power in that, so I think it's great. So again, to show you a little bit more of what Shira has done, we've been developing protein replacement therapies in rare diseases for a long time. I've listed some of the rare diseases for which we have approved therapies, and most of them are protein therapies there, but also I've listed the approximate number of people affected in the world, just to show you that we're very used to working rare diseases, and that dystrophic EB fits very nicely on what we're used to doing. Drug development is different in rare diseases than in other diabetes, cholesterol drugs, and we've been doing it for a long time. So again, dystrophic EB fits nicely. So I mentioned that in 2013, Shira acquired Lotus, and Lotus Tissue Repair, as you may know, was founded to develop a treatment for dystrophic EB using recombinant collagen 7 given by IV systematically to reach all parts of the body. So again, I want to emphasize that everything was a good fit. It's a rare disease, high unmet need, protein replacement, known genetics, and IV. Most of our therapies are IV as well. All right, so I'm going to spend some time giving you an overview of the drug development in general, but also specifically as to where we are and where we have to go. And I think that in general, drug development can be generalized in a simpler way as these 4 bullet points. That you make the drug, whatever the drug is, in a safe and reproducible way. Every time it has to be the same, has to be shown to be safe. And as appropriate, you test the safety and the efficacy in animals, and you have to do that in animals before you give it to humans. It's a requirement. You follow with human trials to test the safety and efficacy, and finally, you seek approval from either the FDA in the US or other regulatory agencies around the world. We've had lots of really good talks talking about these aspects, some of these aspects of drug development, and I hope that my talk will give you a few more details, perhaps, and help with the overall understanding. So the first step, or one of the first steps, is making the drug. And in our case, our approach is to replace collagen 7 systematically. As we've heard many times this week, dystrophic EB is not a skin-only disease. It's internal as well. And so that's our approach. And for this drug development and many others, again, the steps are similar. You develop manufacturing large scale, so you have to make sure that it looks the same, whether you make it in small batches or in large batches. you have to have a process that is very reproducible and controlled. You need to know what you have at every step of the way. And you develop a test in order to do that. And you also have to have a formulation that's stable. Now, we're lucky enough to have a Copeland's and this from DebRA and the Silvers from Jackson Gabriel Silver Foundation come and visit us earlier this year. So I encourage you to talk to them if you want some firsthand account of what they saw at Shire. They talked to a lot of people in our team. They also had tours of the labs and the manufacturing facilities, so it was great to have them. So I'm putting you on the spot, Brett. So as I said, testing in animals is essential, and safety testing is essential. Now, sometimes you have the models, animals that actually have the same disease. And those are great, and they can be used to better understand the disease and give you more confidence in everything that you're doing. Not every time, that's not something you can find every time, but we're very lucky in dystrophic EB that they're great models, and we've made very good use of them and continue to do that to make sure that we know where the drug is, if a drug in animals is going, where it should go, and if it's functioning in animals, how it should function. Now, I have to say that the only way to really know if something works in humans and is safe in humans is to try it in humans. But along the way, you do everything else you can to build evidence and understand as much as you can. So Shire presented some of the results I'm about to show you in May at this congress called the Society for Investigational Dermatology in New Mexico. And I've summarized what we presented in a few bullet points, and then I'll show you some data. So our treatment has the correct structure, and when we gave it intravenously to extremely affected newborn mice with dystrophic EB that have no collagen 7 whatsoever, our treatment went to the correct tissues in these mice. It closed the separation in these mice and improved survival of these mice. Now, these mice only live about 2 days or a couple of more days than that if they don't receive any treatment. And people think that the reason is that they have so many blisters internally that they can't feed. So, so I'll show you some of this data. But before I do that, I thought I would put it very big, what you've been seeing and having to squint at in other presentations, because this will help me actually explain to you what you're seeing. So this is, on the left is healthy mouse skin, and here's where the collagen 7 is. And here is dystrophic EB, the same type of mice that we were working on, no collagen 7. And this is a blister, so the separation where collagen 7 should be. I think Dr. Bruckner called it Velcro, and I think it's a very good way to look at it. So when I show you, I'll be showing you similar slides that where you see either a separation or no separation. And I'll also be showing you other slides where there's going to be some green. And the green is some dye that bound to the collagen 7. And so you'll be seeing green in the middle here, sort of the peanut butter in the peanut butter sandwich. So that's how to focus on. And we have thousands of slides like the ones I'm about to show you. So those are representative, and we've done it a lot. So let me show you some of the things we saw. So this is our treatment given to these mice with very severe DB, and we looked in many, many different tissues. And every time we looked, be it in the belly, the back, the chest, leg, head, and the tongue, and the esophagus, we saw collagen 7 present, even after 5 months. So it went to where it was supposed to go. Then here, we gave it, again, intravenously to these very severe mice. And here is the not treated, and below the treated, same tissue. So in the abdomen, here's the gap, no gap. Back, front leg, tongue, chest, rear leg. So you can see the separation was closed. So, but that's one of the many things we've been doing. It's not the only thing. And I'll show you some of the other things, although I won't have pretty pictures. So drug development really, to me, can be summarized in 2 words: safety and efficacy. And you don't do safety once and you're done with it. You do safety tests every step of the way, and you build on it. So you do it in preclinical studies, that means the animal studies. You do it in early clinical trials, you do it in late clinical trials, and then even after you're approved, especially in rare diseases, you continue to do it, and you do post-approval. Most of our therapies, we're still doing monitoring, and they've been marketed for quite a bit of time. Same with efficacy. So I'll show you in a different way how we do all that. But everything's about safety and efficacy. This is going to look complicated, but I think the take-home message will be fairly simple. And it is that there's a lot of things that need to be done in order to get a drug approved, but that all these things are done. Some of them you can only do in a certain sequence, but others you can do in parallel, and whenever possible, that is what we do. And we work in parallel when we get ready so that we're ready to go as soon as possible. And there's some very defined moments in the drug development pathway, and I think you already heard about the IND submission, which is, actually let me advance one more. So IND means investigative new drug, and it's an application to the FDA or some other regulatory agency where you put together everything, a company puts together everything they know about the safety and efficacy and how the drug is made, and you go to the FDA or some other regulatory agency and you ask basically permission to go into humans. So after that, so actually before that, let me just tell you where we're at. This is the very beginning of the drug development. This is the basic research that you've heard about and that is done has been done in so many labs, and you heard from so many people who have done that work. Understanding collagen 7 and understanding the gene and all that. This is where Lotus was, proof of concept in an animal model, meaning proof of concept, proving that it works. We went ahead and repeated that quite a bit, and we've been working on all of these things, and I'll show you in another slide about orphan drug designation, about natural history studies, but we're also working on manufacturing for protein, and that's part of what the labs that Brett saw. You start with a small batch, and it gets bigger and bigger till you have literally a vat that's as big as a room. And let me pause and tell you that the way we make this protein is in cells, not cells that get implanted in your body like you've heard, but cells that grow in a vat and produce collagen 7, and we purify that collagen 7 and put it in tubes. So different from some of the things you've heard this last few days. And doing some of the animal studies, the safety studies, the pharmacology, pharmacokinetic, and all that. And we're also starting on natural history studies. So we're working on all these at once, as well as repeating animal studies to learn more about dose, to learn more about what's called dose regimen, which is, how much do you give and how often. And they're models, and you can use animals to figure out what you think and get an approximation and put it all together and go to the FDA or the EMA. And then after that, if they say okay, then you start your clinical trials. Now, the next slide is going to show all that, the clinical trials in more detail. So let me go to that. And something to emphasize here is this is really a model of a clinical pathway. Most of the time, it doesn't really look like this. It might be that you could have a phase 1 and then a 2 and then a 3, or you could have a phase 1/2, then a 2/3, or other variations. What always happens is that phase 1 has to be before phase 3. So there's certain steps. And I think yesterday, if you heard my colleague, Dr. Charnas, he gave a really nice presentation of the clinical pathways. So this will be a little bit of a review. But I think the take-home message is that at any time, even before you go to the FDA for an IND, you can do a phase 0. Phase 0 means it's observational. You're not giving treatment. You're trying to learn about the disease. And they're also called natural history studies, observational studies. Then phase 1 is primarily about safety, but sometimes it's a phase 1/2 where you're also trying to get efficacy. Does it work? Is it safe, and does it work? Eventually, you build, all is checking safety, all is checking efficacy, and you build a package that again, you submit to a regulatory agency and you say, are we allowed to market this, to approve a drug? But it doesn't stop there. We continue to do, especially in rare diseases, to do phase 4, which is monitoring for safety, efficacy, et cetera. Let's see, I think that, why do we do natural history studies? And this is something that is becoming more common in all drug development, but it's always been more common in rare diseases, and it's extremely common now. Rare diseases are poorly understood, and because of small populations, few studies, but it's so important to understand the disease so you can build your clinical trial and you can understand the changes. If you give treatment and you see a change, and if you don't know how the disease progresses, you don't really know if the change is due to your treatment or not. So having those studies done it's really essential. So what do we learn? We better understand the disease characteristics, we understand the progression, and you may get some information about tools to measure. So just saying that, oh, I feel better or it looks better is not good enough for regulatory agencies. You need to measure, and numbers and any other way you can show and do statistics on it. So, and that's often very difficult. The other thing you may be able to identify is biomarkers, if you're lucky, things that change and are easy to measure but not related. But mostly you want to avoid pitfalls that will slow your drug approval. You don't want to be told, you don't have enough data, you have to go back, do more. You want to really do everything you can ahead of time. And so it's a way to be efficient as well as to understand the disease. And for dystrophic EB, Shire has really started a natural history study earlier than we've ever done in the past before. So we've broken it into 3 steps. First, the assessment and optimization of imaging technique, and we used normal volunteers to do this, and that's done. How to take pictures and also some more advanced ways of doing imaging. And another one is ongoing. We're calling it the STOS. So it's several sites in the US and around the world, but also we're looking at relatively small number of patients and a few visits, but we're measuring wounds and we're doing existing questionnaires, and we're doing everything and anything that is out there to asking questions to try to get the data. We're also developing our own instruments, or at least parts of it, and we're going to take all that to the FDA and say, this is our proposal, we've looked at it, do you think it's okay? So this partnership between companies and regulatory agencies is something that we're very used to and is very good in rare diseases. You know, it's really a group effort. The patients, the patient organizations, the physicians, the companies, regulatory agencies, all working together, because all of this is new to everyone, and we need to figure it out together. And finally, we're going to take all these results and build a natural history study that will go probably for a long time. We'll follow people over time to really understand the disease. So that's planned in the short term. So as I promised to say, we did receive orphan drug designation for our treatment in the US in March and Europe in June. But I want to emphasize this is one of the many steps. This doesn't mean that we're ready to put it in patients. That's not what it means, but it's still a very important step. Finally, I want to thank you again for giving me the chance to speak, and to tell you that Cher has always worked with patient advocacy groups and patient groups and patients, and we really do respect your knowledge and expertise. We're the experts in drug development and protein replacement, but you are the experts in DB and EB. And we're committed to working with patient organizations that have been working to bring therapies as quickly as possible to patients. So, and finally, thank you.